All right, let's dive right in. Today, we're talking about a landmark study from the New England Journal of Medicine, and it details what really looks like a functional cure for sickle cell disease. This is one of the world's most common and, frankly, most painful genetic disorders. And this isn't some kind of science fiction. This is a one-time treatment that's changing lives right now.
To really get why this is such a huge deal, let's just start with a question. What if your own body was a battlefield? I mean, for millions of people with sickle cell, that's not some metaphor. It's their daily, painful reality, where their very own red blood cells become the enemy.
So, what's it actually like to live with this disease, day in and day out? Let's break down the sheer burden of it. For patients with the severe form of sickle cell, life is often just measured from one crisis to the next.
And the thing that defines this disease is something called a vaso-occlusive crisis, or a VOC. Just imagine your red blood cells, which are supposed to be flexible and round, suddenly turning rigid and shaped like a crescent, like a sickle. Well, they get stuck in your tiny blood vessels, and they create this logjam that blocks blood from flowing. The result? Sudden, excruciating pain that can pop up anywhere at any time, and it's so severe it often means a trip to the emergency room.
Now, this slide is what really puts that suffering into perspective. This is the baseline data from the actual patients in this study, before the treatment. On average, they were dealing with about 4 of these severe pain crises every single year. And look at that second number, almost 20 days a year spent in a hospital bed. That's nearly 3 weeks of your life every single year, just stolen by this disease.
So, how do you even begin to fix a problem that's literally written into someone's DNA? Well, it turns out the answer was also written in their DNA the whole time, a kind of hidden solution just waiting to be unlocked.
And this here perfectly lays out the problem and the solution. On the left, you've got that sickled adult hemoglobin that causes all the blockages. But on the right, you have fetal hemoglobin. It's what we all have when we're newborns, and it's perfect. It's round, it's efficient, and it doesn't sickle. The problem is, our bodies switch it off not long after we're born.
So this is the crucial piece of the puzzle, a gene called BCL11A. The easiest way to think about it is like a genetic light switch. When we're babies, that switch for fetal hemoglobin is flipped on. But as we grow up, BCL11A comes along and flips that switch off, and adult hemoglobin takes over. Now for people with sickle cell, that's the moment the problems start. So the big question for scientists became, hey, could we find a way to flip that switch back on?
And that question brings us to the treatment itself. It's called exa-cel. This is where truly cutting-edge technology comes in, to literally rewrite a patient's own genetic code. Let's walk through how it works.
The tool they use is the revolutionary gene editing tech CRISPR-Cas9. Really, the simplest way to think about it is like a pair of molecular scissors. You can program it to find a very specific sequence in your DNA, in this case that BCL11A off switch, and just make a super precise cut.
The whole process is pretty remarkable. First, doctors collect a patient's own blood stem cells. Second, those cells get sent off to a lab where CRISPR is used to snip and disable that BCL11A off switch. This basically forces fetal hemoglobin production back on. Third, the patient has to go through chemotherapy to clear out all the old, unedited stem cells from their bone marrow. You got to make space for the new ones. And finally, those edited cells, the exa-cel, are infused back into the patient where they get to work producing brand new, healthy, non-sickling red blood cells.
So, did it work? This is the moment we've been waiting for. And the results from this clinical trial are, honestly, nothing short of breathtaking. This is what a functional cure actually looks like in the data.
Get this. Out of 30 patients with enough follow-up time, 29 of them, that's 97%, were completely free of those debilitating pain crises for at least a full year, and many for much longer. Just let that sink in. They went from an average of 4 severe crises a year to 0.
And it gets even better. 100% of those patients were completely free from hospitalizations because of those crises for at least a year. They went from spending nearly 3 weeks in the hospital every year to not at all. The impact that has on their lives is, well, it's almost impossible to overstate.
And this chart right here shows why it works so well. Look at the percentage of that protective fetal hemoglobin. Before the treatment, it's just this tiny fraction, right? Around 5%. But just 6 months after the treatment, boom, it skyrockets to nearly 44%, which is a level known to be super protective. And it stays there. The switch was successfully flipped back on and it stayed on.
This is a quote taken right from the study. And it's so important because it's not just about the numbers and lab values. The researchers measured quality of life, pain scores, general well-being, and across the board, patients reported huge, clinically meaningful improvements that got their lives back.
This really is a new chapter for medicine. But, like any major breakthrough, it's really important that we understand the whole picture, the challenges, and the road that's still ahead.
So the treatment process itself is incredibly demanding. This slide shows the most common serious side effects, and they are pretty significant. But here's the crucial context, and the study makes this clear. These side effects are what you'd expect from the strong chemotherapy you need to clear out the bone marrow. They aren't thought to be caused by the edited cells themselves. So yeah, it's a difficult journey, but it's one that leads to a really profound reward.
So the ultimate takeaway here is just monumental. We're looking at a one-time therapy that for pretty much every patient treated effectively eliminates the single most defining, agonizing symptom of severe sickle cell disease.
And that leaves us with one last profound question. This treatment proves that we can go into our own cells, find a single genetic instruction, and literally rewrite it for the better. If we can do this for sickle cell, what's next? Is this the beginning of the end for genetic disease? The journey is far from over, but for the first time, it feels like the destination is truly in sight.